Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Dr. John Eiler. Listeners, John is the Robert P. Sharp Professor of Geology and Geochemistry, as well as the Ted and Ginger Jenkins Leadership Chair of the Division of Geological and Planetary Sciences at Caltech. And today we are looking forward to discussing his career and his work on a PCC-funded research project investigating the use of molecular isotopic structure as a tool for doping forensics. And we'll break that down in our conversation today. But first, John, welcome to the show. How are you?
I'm very well. Thank you very much for the invitation, and I'm really looking forward to the conversation.
We are thrilled to have you with us, and perhaps to give our listeners a little bit of background here, can you just start by telling us more about yourself and maybe how you got interested in geology and geochemistry?
I grew up in Madison, Wisconsin, which is a university town, but in the 70s and 80s when I was young, it was also kind of a cultural hotbed with first wave punk bands and all kinds of interesting things that would catch the attention of any teenager. I went to Beloit College in southern Wisconsin, an ACM school in the Midwest. And initially I thought I would study history, but I was not good at reading and writing, so that wasn't going well. And I was taking geological courses because they were the easiest STEM course I could take, which as a geology professor, I will admit, this is basically true, at least superficially. But my professor at the time, a man named Hank Woodard, who was teaching me introductory geology, he realized that I was kind of big. I lifted weights every day, which we can come back to because that's relevant to our topic. So I was obviously could pick things up. And he knew just from talking with me, I knew how to fish and things like that. So he hired me as a porter on his geological excursions, and all the students and he would go out all day and make maps and collect rocks and talk about them back in camp. And my job was just carrying canoes around and fishing and cooking for people, cleaning camp and things. But I just fell in love with the lifestyle, the vibes. It just all felt so much more natural to me as a way to spend your day than other things I was doing in school. So I steered into it and somehow never stopped. And now here I am.
Well, I think that is a beautiful origin story there. You're the first one I've met who kind of stumbled into science in this sort of way. So can you talk about then what steps you took from this initial porter position to get to this position you're in right now, this distinguished faculty role at Caltech?
It's not any sort of intentional or linear path, and it's different from the experiences of most of my colleagues that I interact with day to day. When I was a high school student, I think I was clever enough for cracking jokes and things, but I was not an accomplished student, and I was not an accomplished student in the first year and a half as an undergraduate. But I married young, and I met my wife when I was 18, first year of college, and followed her to Iowa City when she transferred there. And that was where I started getting serious about academics, mostly because I didn't know what else to do with myself. My wife was very scholarly, and I spent all of my time lifting weights and hanging out with friends and drinking beer and stuff like that. And I couldn't do that in Iowa City. I didn't know anybody. We lived in an apartment far from the social hub of campus. And I had to go to class anyway. And I was in this new environment at a big state school taking remedial STEM classes with the pre-med students. And there'd be 500 kids in a class. And I just had the thought almost out of the blue, why don't I just do this? I'll compete with them initially. I'll see, can I be better than the student who thinks they're the best? And just try that on for size to keep my mind busy. And it kind of worked. I discovered I was good at it. And initially, I was just rewarded in simple ways. I'd get a good grade. People would say I was doing the right thing. And that just sort of drew me into this lifestyle of focusing on my studies and being academic. And then field experiences, research experiences really made this connect to me personally and made me realize it was something I wanted to do as a job. And that just grew over graduate school. There never really was a point where I thought, this is a thing I absolutely have to be doing in just this way. It wasn't a thing I was doing when I was a young child. I just sort of drifted into it. I would say to this day, if I woke up tomorrow and you said, John, you're now a professor of Byzantine history, you're not a scientist anymore, I would say, okay, I'll do that. I'll try that on for size and see if I can make it work.
Well, I love that. So let's talk a little bit more about what you do there at Caltech right now. Can you describe your current role and maybe share the scope of the research that you do in your lab there?
At the moment, my title is chairman, so I run the Geological and Planetary Sciences division at Caltech. But an important thing about Caltech is it's so small and it's so focused on research and so kind of flat, I would say, in terms of organization, that everybody here, from the president, the provost, the chairman of departments, we're all working scientists. We all run labs, supervise students, write papers. To the extent that there are 24 hours in a day, we keep our career going through those sorts of title changes and new administrative roles. So my research group, it's a kind of idiosyncratic thing. My view is that every new person, every postdoc, every graduate student who drifts into my lab group, they're all individuals. They all have their own passions, their own strengths, their own wishes for themselves in terms of their career. So you don't really know what they should be doing, how you should be helping them or guiding them until you've gotten to know them. And therefore, because they're the ones doing most of the work, you don't know what your lab's going to be doing. It's a thing that just evolves in response to who shows up. But there are some core themes. One of them is technology development. So I just love the concept of observing new things, discovering new things. And one of the main ways that natural scientists do that is to think of a new way of looking at things, think of a new way of observing nature. And out of that will come discoveries. And you'll just use the scientific method and your own instincts to follow them up and just trust that something good will happen if you observe in a new way. So the way that that gets expressed in my group is through inventions of new objects, new technologies, mass spectrometers for observing the isotopic contents of natural materials of all kinds, and then inventing new methods for using those technologies, inventing new concepts for how we talk about the way naturally occurring isotopes are distributed in nature, and then connecting all of those things to big questions in the natural sciences and the fundamental sciences, and in some cases, engineering sorts of problems. It sounds very broad. It is very broad. Some people in my group right now study the origin and evolution of the early solar system. How did planets form and from what? What were the chemical processes happening then? Other people in my group are studying the way that chemistry organizes itself to enable the emergence of life. How does organic chemistry in the dead world around us turn into life? How did that occur? It obviously happened. We're sitting here talking, but we don't know how it worked. So there are many sides to that problem, and my group contributes to some of them. Other people in my group are studying doping forensics and other biomedical and environmental problems. That's why you and I are talking today, and on, and various other problems as well.
I think that's really cool. And I think having this broad portfolio of research in your lab really makes sense because the technology and the methods that you work with are applicable to such a wide variety of different scientific fields. And I know over the past couple of decades, you've been collaborating with Thermo Fisher Scientific actually to make some important changes to adapt mass spectrometry technology specifically for your work. And I'd love to dive into some of the details there. This started way back in 2003. So John, can you talk about maybe first the scientific problem that you were trying to solve with this technology and why it was so important to you?
This harks back to what I would say is the turning point in my career as a research scientist. I had a productive, but relatively speaking, conventional research career as a student and postdoctoral fellow. And for the first couple of years as a faculty member at Caltech, I was using methodologies that were cutting edge, but known. Other labs were doing them. They had a prior history. The concepts were relatively established. And I was just using them as best I could and in the most creative ways that I could. And then around 2003, I was coming up for tenure, which is sort of a watershed experience for academic scientists. And I asked myself some big questions. I felt like I had done enough. I had crossed whatever the threshold or bar was to continue in my career. But had I done enough for me? Was I really being the scientist that I wanted to be? And I thought deeply about the sorts of scientists that I most admired and that I most related to. And there's many different ways of doing this job. Some people become incredible scientists by really focusing on one big question or one big problem and just dedicating themselves to it over decades. That's not me. That's not really my spirit. And I knew that about myself. And so the scientist I was most meditating on was a fellow named Sam Epstein, who was a senior professor in my department. He was actually my first postdoc supervisor at Caltech. And he was this very different kind of scientist. In personal presentation, you might have found him kind of wandering in his thoughts and language. He would contemplate any kind of problem. He was curious about every kind of problem, very fearless in his commitment to attacking problems of all sorts. And because of this, he founded many different subdisciplines of geochemistry, that is the study of the chemistry of the natural world. And what I loved about him is that he was just an explorer. Every day he would wake up and go somewhere new that he had never been and no one else had ever been. So I asked myself in 2003, how do you do that? Is there anything new? Young scientists often face this problem. They look around themselves and there's so many accomplished people, so many technologies, so much infrastructure, funding programs. There's so much going on that feels very structured and organized. What else could there be? What is outside of that? So I really wanted to address that question. It felt very personal to me. And I just had a gestalt from reading at the time. This is sort of a shaggy dog story, but it goes somewhere important. At the time I was working on molecular hydrogen as a fuel. This was a period of hydrogen economy craze that I got caught up in and I was studying. And to apply the field of isotope chemistry to it, I was reading these very old papers from the 1930s from a scientist named Harold Urey, who basically invented isotope chemistry as applied to natural things. And he was interested in the form of hydrogen that had two rare isotopes in it. And I looked at that and I thought, that is so weird. No one studies that. No one studies molecules that have two rare isotopes in them because they're so rare.
Right.
So Urey thought they couldn't be studied in nature. No one else appeared to have thought they could be studied in nature, and yet they exist. So I thought they're out there. And then when you think about it, you realize just through the combinatorial math of how a rare thing can fit into a big complex molecule, maybe there's molecules with three rare isotopes. Maybe the 2 or 3 or 4 or more, maybe they're positioned in different parts of the molecule. How many ways could that happen? Oh my goodness, many, many, many. There could be thousands of forms, millions of forms. And I realized almost all of the diversity in the isotopic content of nature is recorded in these very rare things, and no one has ever seen a single one of them. Not once. Not in any meaningfully precise way that they did it on purpose. And so I thought, I'm going to be the one who does it. I'm just going to start trying to do that. And I initially did it by hacking instruments that were already in my lab, conventional mass spectrometers, to create these very poor precision and yet just barely useful measurements of rare isotopic forms of carbon dioxide. And then I just pulled that thread and pulled that thread and pulled that thread, leapfrogging from one new instrument design to the next to the present day, where we basically, I would say today, you could name any molecule known to science and say some very weird isotopic version of it. And unless you were being a jerk and picking something impossible, I would design a measurement we could do in my lab, and within a few hours, we would be trying it. That has been the arc of my career. And I just trusted that if I do this, if I go this place that I haven't been, there will be good science there. Questions will arise, new attacks on big problems will become possible, and I'll just steer into them as I recognize them.
I think that is remarkable. And I have to ask, how did the people in your field, or perhaps the people you were collaborating with at Thermo Fisher Scientific, respond when you brought them this proposal of what you wanted to do?
It's a little bit of a shaggy dog story, but it's such a beautiful relationship, and it's been so productive over the decades now. I just have to tell you, the first thing I did is I just went by myself into the basement and I started playing with my conventional mass spectrometers, hacking how they were being used, using them in a way like people used mass spectrometers in the 1930s and 40s. to force them to do new kinds of observations, to study these very rare ion beams, ionized versions of the molecules I wanted to study. And then I realized I can't go on like this. I need to create instruments that are designed for this purpose, and they should begin as close as possible to the ones that already exist so that it's practical and I can make progress quickly without overreaching, and I can start to learn things quickly and just let the whole thing evolve naturally rather than trying to jump to some end solution. So I started calling all the different companies that make scientific mass spectrometers that are suitable for this purpose. And I knew a bunch of these companies. I ran an ion microprobe lab that had instruments from another company. I had colleagues who used instruments built by companies other than Thermo Fisher. Everybody said no. They said, there's no market. It isn't obvious this is going to work well enough to teach you anything. There's no application that anybody knows. So no, it's not worth it. We can't invest our R&D time into something like this. Thermo Fisher, they just had a different attitude. And I think it may be because they had a decades deep culture going back to the 1950s, even before, of mentorship of their own staff. They have almost a kind of apprentice guild-like system of training people and retaining people, and a commitment to a long-term vision of building out what can be possible, what can be done. And so they took it on. And I wrote a proposal to them. I said, hey, I want you to build me a machine that's like, does this and this and this. And they said, great, let's do it. And I went, raised some money, paid them the check. And then they did something, let's just say it was good enough. It wasn't quite what we had in mind, but they delivered an instrument that was just barely capable of letting me make a good quality measurement of rare isotopic forms of a few simple molecules, simple gases like carbon dioxide or oxygen, or things like this. And I said, I'm not going to be a nitpicker. You didn't build what we said we thought we'd build, but you built something that works. I'll just go use it and I'll get back to you and let you know how it's going. 2 years later, there was a couple of big breakthroughs. We figured out how to observe these very rare multiply substituted, that is, molecules that have multiple rare isotopes in them, forms of simple gases. And we figured out how to measure them really surprisingly well, very precisely. Did a few basic science studies of how they behave, and then quickly, in consultation with various colleagues of mine who are paleoclimate researchers and geobiologists and things, I started a program studying how this sorts of rare isotope forms behave in old fossils, in limestones, and things from the geological record. And this application let us measure the temperatures of formation of all kinds of things like dinosaur teeth and 100-million-year-old eggshells from bird-like dinosaurs. And so many different things became possible very quickly. And a whole community just exploded that wanted to adopt this and start studying this. But I looked at it and said, that's great. I love that energy. I love all these people showing up. All of them want to do something that I just did a year ago. I don't want to do what I did a year ago. I want to do what I'm going to do a year from now. I want to do something new. I want this to grow and build and expand until we're studying everything this way, not just carbon dioxide or another simple molecule. I want to study everything. So I went back to Thermo Fisher and I didn't tell them I want to study everything. I told them I want to study the next most complicated thing, simple little organic molecules like methane or things like it. Simple little gaseous molecules that have carbon and hydrogen in them in organic kinds of chemical bonds, because most of chemistry is organic. Think of every molecule known to science, 99% and more of them are organic molecules. If I can't study them, I can't realize my vision. So we then designed a much more ambitious instrument that was capable of this. And this time they really went for it. And they not only met the design goals that we discussed, but they exceeded them. And then they designed a whole new platform and they spread those technologies, elements of them, to other instruments that they build. They really committed to it. And out of that came an instrument that gave birth to another subdiscipline, people studying natural gas, biology and the environment, all kinds of problems. Abiotic chemistry, that is organic synthesis without life that happens in the crust of the earth. Many different subdisciplines were attracted to this, and that's great. Everybody's welcome. All these new disciplines are welcome. All these new people are welcome. I love that. I'm moving on. I'm going to go to the next thing. So this was a really critical threshold for the explosive expansion of this field. When working with the Thermo Fisher team, we figured out how to pivot all of this science, all of this technology from mass spectrometers that are, I would say, relatively conventional. We don't have to get into all the nuts and bolts in this discussion, but they perform a kind of obvious task. You take molecules and you literally sort them in space so that all of the ones of one isotopic form go in one space and all the ones of another isotopic form go to a different space. And then you count them. We were never going to get to where I needed to get if this was how we kept going, because your capacity to spatially separate molecules into different locations, it's just limited. You bump up against all kinds of technical engineering problems. There was another way, another path, and that was to adopt the technology of a thing called the Orbitrap. It's unique to Thermo Fisher. It has concepts that are related to other kinds of technologies that other companies build, but nothing like their scale and cost. Much more flexible, much more suitable for experimentation and design of new measurement concepts. The Orbitrap is basically a little metal football the size of your thumb. It looks like nothing. You can't believe you're even looking at anything that's useful at all. But it was designed by a genius, a guy named Alexander Makarov, who works for Thermo Fisher in Bremen. He invented this thing, and it basically allows you to take a group of molecules, put them into a little teeny cavity, and make them go in orbits, little circles, around a spindle in the middle of that cavity. And you don't ever separate them. They stay together the whole time, doing their little orbits, and you just listen to them. Literally, you have a kind of antenna that is listening to these molecules spin around, and then you do some fancy math on the sounds that you heard, in a way, when you listened with your antenna. And from that emerges a statement of all the different isotopic forms of that molecule that are present with just exquisite separation, and many, many of them observed at once. So this was the breakthrough thing that allowed us to go to steroids, what we'll talk about today, and many, many, many other kinds of organic molecules and inorganic molecules, basically everything.
Basically everything. The goal was realized, it sounds like.
You got it. It's a funny feeling, but we set an impossible goal and then somehow we're all there. I don't know why we're there, but somehow we're there.
Well, John, I think this is amazing. And I love that you've been just a huge trailblazer in the field where you just kept pushing those boundaries. And I know the Orbitrap that you talked about is widely used in anti-doping labs today. So I'd love to dig into this anti-doping connection. Can you share how you first found out about the Partnership for Clean Competition and maybe realized the work that you were doing was relevant and then ultimately decided to apply for a research grant?
There's so many different sides to what you just said. Let me touch on them sort of in the order in which you mentioned them. Mm-hmm. First of all, you say this technology, all the doping labs already use Orbitraps. Exactly. You're right. And this is very important that they already use them. But there's a critical difference. They're using them to identify molecules. The hack that we did is to say, yes, now you know what the molecule is. What's next? What else can you ask about the molecule? So we're taking an equipment that already exists in doping labs, and we're hacking the way that it's used. We're hacking the way samples are introduced to it. We're changing the way that you measure so that once you've identified the molecule, now you can ask many higher level questions about where it came from. What has it experienced? How was it made? So this connection between what Thermo and I had been working on and what the doping community was doing, it's very close. We're just automatically right next to each other in terms of the technologies that we're using. We just need to figure out how to use them differently. So why would I approach the Partnership for Clean Competition to try and make this transition, to get us all using these technologies in this new way? Well, first of all, there's a kind of deep motivation. I've mentioned a couple times, I kind of grew up in weightlifting gyms in the 80s and early 90s. If you saw me, you wouldn't realize it. I'm just kind of a normal person because I don't dope. All right, there we go. I use the hormones that the universe gifted to me, and they were not very special. But I liked being in gyms. I liked lifting weights, and I was around all kinds of people. And everybody who understands this community knows it's not just the top competitive athletes or the bodybuilders you'd see on TV. Many people in gyms who are just ambitious and they want to get bigger and things, they dope. It's a common thing. I never did it, but a lot of people did it. So I was familiar with the existence of this subculture of doping. And I always had, I wouldn't say a conflicted view, but like two sides to the coin. I don't mind people putting whatever they want in their bodies. It's your body. Do what you want with it. But I don't like liars. Somehow that bothers me. The idea that somebody would stand in front of a camera and lie about what they're doing, that I don't like. So it always stuck in my mind. People should just be honest about what they're doing. And if there's rules in a space, you should follow them. And if you're not following them, you should say so. So I always felt that way, although I never had any particular thought about how you'd scientifically approach it. But as I grew as a scientist, I was aware there's an entire science of not just detecting that people have elevated levels of hormones in their bodies. Of course, that's an obvious thing to check. You can read about that in the newspapers. But there's this other side. What do I do when people are tricky? What do I do when somebody has doped, but they're smart about masking it or timing it or flushing it from their system? And the moment that really caught my attention was the Floyd Landis case that anybody interested in this podcast will maybe remember, all the press about Floyd Landis. But I'll briefly recount it just so the story is in everybody's mind. So Floyd Landis wins the Tour de France in a spectacular fashion, one of the last days. And everybody's amazed. Oh my gosh, Floyd, how did you do it? I don't know. I just thought I was inspired. The next day, the news drops. He's positive for steroid doping. And his immediate answer is, well, I just have a lot of testosterone in me. That's who I am. Okay, nobody believes that. But it's a defense. You can say it. If you want to say it, you can say it. And then he said a million other things. Oh, I drank whiskey the night before, or I ate a hamburger, or just a bunch of nonsense. But how are you supposed to disprove his nonsense? And then the hammer dropped and he got busted for real. And it was an isotopic test, a test that I understand because it comes out of my field. It's a much simpler thing than what my group does, but it worked. What they did is they showed that the carbon-13 content, a rare naturally occurring isotope of carbon, was different in his testosterone as compared to his other hormones. And it was different in a way that made a lot of sense because synthetic testosterone at the time, maybe still, is made from phytosterol precursors, a plant sterol that mostly comes from soybeans. Soybeans are different in their carbon isotope content than the average of all of the food a human eats. So the carbon you use to build your own hormones has different carbon-13 content from the carbon used to make what you inject. And he just couldn't avoid it. So I followed this and I just sort of packed it away in the back of my mind. And then one day I opened my email and I saw an announcement that we get weekly at Caltech of funding opportunities. And I'm just kind of scanning down and I see one, Partnership for Clean Competition. Okay, I didn't know that that was a thing, but it sounds interesting and it sounds related to all of these interests I have bumped up against in my life, things I remember doing and seeing and talking to people about. And then I read the call and I saw, well, they're interested, maybe they'll do other things, but they're interested in pilot projects for very new ideas. And I thought, well, that's my lane. Mm-hmm. What if I could take these novel technologies that we'd been inventing in my lab and apply them to the doping problem? And specifically, the thought I had is Floyd Landis got busted because it probably never occurred to him. If you're going to stick testosterone in your arm, you better have the same carbon-13 content as your body, or you're going to get caught.
Right.
So if you did this today, if you'd read the newspaper and you knew what happened to Floyd Landis, unless you're a dummy and you know you're going to get tested, you should say, you know what? I'm going to go find some or make some that has the right carbon-13 content, and then it will be invisible to all the known tests. I knew there's nowhere you can be invisible to my test, because my test has access to every isotopic form. That is, not just how much of the isotope is present, but where is it in the molecule, atom by atom? Are there two rare isotopes in the molecule? If there are, where are they? If you think this way, you'll quickly realize there's hundreds of different things we can measure, each of them independent statements of how that molecule was made. What was it made from? How was it assembled? Impossible to mimic. It would be like copying somebody's thumbprint down to every level of detail. You're never going to do it. So I thought, I'll bet something like that is possible. I will write to the Partnership for Clean Competition and say, let's start this. Let's see if we can make this physically possible with a little pilot study. And that's what we did.
I think that is so cool. And I like that you took that leap and thought, I'm not sure what they're going to think about this geologist, this geochemist reaching out and saying, hey, I want to do anti-doping research. But I think that's exactly what the Partnership for Clean Competition is looking for, these multidisciplinary collaborations and projects to bring new technologies and new techniques into the field of anti-doping. So I think that's perfect. And you touched on this, but just knowing the molecular structure of isotopes can open a wide range of detection abilities here. We've been focusing on testosterone, but perhaps could you maybe comment more broadly on how this would be valuable in the context of anti-doping as a whole?
The way that we pursued this, first of all, I know what I'm capable of. I also know my limits. I am in the end just a geologist. So I knew I needed friends to help me do this. So I called around and found a wonderful colleague named Thomas Piper, who's at the German Sports University in Cologne. And he's been working very closely with us. He'll co-author the paper that we're submitting. I also knew I needed a person in the lab who really understood not just one compound like testosterone or something, but all of the metabolism. Somebody who understood the biochemistry of how all of the steroidal compounds relate to each other, the entire reaction network that is being perturbed when you dope with some steroidal agent. So I found a great young scientist named Lubna Shawar, who at the time was a postdoc at MIT, but I hired her to come to Caltech specifically to work on this project because I immediately had the thought, I know what testosterone is. Everybody who reads the newspaper knows what it is, and they know that people dope with it. But I also knew just enough biology to recognize it's bigger than this. Really, what you want the capacity to do is to freely assay all of somebody's steroidal chemistry and recognize when it has been perturbed. Maybe they do something very simple and obvious, like injecting testosterone. Maybe they inject something else that promotes testosterone. Maybe they inject a synthetic version that mimics it. There's many different compounds that could potentially be injected. And the arms race in doping and detection is very much about this problem. The fact that I can achieve the same outcome in a variety of different ways. And simply detecting the presence or absence of one molecule, that's never going to be a permanent solution to this detection problem. You need to be able to sort of freely observe the entire system and notice when something has changed in it. So one of the questions that we had at the outset of our study in conversation with Thomas and with Lubna was, can we notice not only that a person has put something exotic into their body and then you watch it being there and then slowly diluting away? That's a very obvious thing. Can you also notice the perturbation, the change in the metabolism of other hormones in their body because they doped? And this is a very subtle point, but it's one that ends up being quite important in terms of understanding what we finally observed in our study, that if I were to inject one hormone into my body, you could watch its concentration rise and then slowly fall as it goes back to whatever the natural abundance would be. But that isn't the only thing you've done. It's sort of like a hammer hitting a bell. The drug you injected, that's the hammer. But the bell you hit, that's your whole metabolism, and it's going to ring for a long time after you struck it. So can we see that? Can we document it? Can we understand it? And now have a much more specific and nuanced way of understanding what has occurred in somebody's metabolism, which is exactly what we ended up seeing. So our goal all along has been to figure out, could I observe one or more or a bunch or maybe dare to dream all of the active hormones, the androgens and other related hormones and compounds in your body, look at all of their isotopic structures and see how the entire system is behaving. Is it acting in a naturally regulated way, or has it been perturbed in some exotic way?
Definitely. And the way you describe it, it all sounds so easy, so straightforward. You just have to do A, B, C, D. But I know that's not how science works, right?
Yeah.
What have been some of the challenges that your team has encountered while working on this particular project? And maybe can you talk us through that and how you overcame some of those challenges?
So you have a number of challenges. First of all, you always have to respect when you go into an analytical lab, if you're doing a measurement that is relatively new, maybe challenging, then you have to respect every step of the process, even things that you think of as routine. And in this case, we wanted to measure a variety, about a dozen, of different isotopic forms of hormones from urine samples. And we had to, first of all, stop ourselves and say, okay, we're about to make a measurement that as recently as 3 or 4 years ago, anybody in the world who's an expert of this would have said is physically impossible. They would have said this will never be done. We know it can be done, but it's only because of things we've recently done. We have to keep pushing on all the fundamentals. Every piece of it has to be done perfectly. So one key part to this was just the isolation and purification of each compound. And for this, we relied on Thomas Piper at Cologne because he's been doing it for many years. He has a laboratory specifically built for this purpose. And we knew if you need to fix your car, go to the mechanic. Don't do it yourself. Have somebody who knows what they're doing do this. Maybe I should have compared him to a brain surgeon instead of a mechanic, but these are all worthy jobs. So hopefully he'll understand what I meant. And then the second thing is standardization is absolutely essential. We are doing something that is so incredibly subtle. If you were to look at what we call the mass spectrum, that is the distribution of masses of all the versions of the molecule and all the pieces of the molecule and their abundances as we measure as intensities in the analytical instrument, if you were to look at a sample that was very exotic to us and another sample that was, let's say, normal, a normal natural hormone sample, just with the naked eye, you would say these are identical. In every way, they look identical. It's because the changes are incredibly subtle. They're fractions of a percent of the height of a tiny, tiny peak that's almost invisible in the mass spectrum. So doing this correctly, you need to introduce the specimen in a very controlled way, compare it to a reference material that's introduced in a similarly controlled way. You have to have very finely controlled ways of separating, isolating parts of the mass spectrum. And then you have to watch it under uniform conditions for literally hours. Whereas a normal scientist just detecting these things would look at them for a few seconds just to notice that they're present. So honestly, the way that you make this work, it's a crazy concept. I'll acknowledge the idea of doing this is way out there. The mundane reality of doing it comes down to just hard, detailed work in a million little steps. So the only people who can pull it off are super meticulous lab scientists. And Thomas is like that, and Lubna is like that. So basically, I found Thomas and Lubna, and then I cheered and clapped at them and congratulated them at the end of every day. And
that's an important role as well, I will say.
Yeah.
And I know, John, you have a paper in the works for this project. So can you maybe share with us some of the results that you've gotten to date?
I would say it's very exciting as a proof of concept. And as a research scientist, I look at it and say, what I love most about it is what it tells me to do next. It's clear that there's some new possibility of what should come next. But let me tell you what we did. So Thomas has a laboratory that has access to all kinds of samples that are both things taken, let's say, out in the wild, let's say at a sporting event that are subject for testing as part of some sports anti-doping program. And then he also has controlled experiments he conducts. So we decided, let's begin with something where we know what we're looking at. So he took 2 subjects in the end, and he had one of these fellows drink a solution of water and ethanol that had been spiked with the steroidal compound androstenedione, which is part of the complex reaction network that creates and consumes testosterone. And the other man was given a similar solution spiked with testosterone itself. And then Thomas collected urine samples from these subjects over the course of the following 2 days, and he isolated out of them the compound androsterone, which is one of the steroidal compounds that can be generated from both of these administered compounds, but through different pathways. And then we watched what would happen to the isotopic composition and concentration. And first of all, we saw exactly what you might imagine in terms of concentration. If you stick something in your arm and squirt, well, then it's going to spread through your body within minutes to hours, and then your body will flush a lot of it out and metabolize it, turn it into other things, and the concentration will drop. And it does so in a time period of sort of half a day. And then we had the conventional isotope data, where you just count all the rare isotopes of carbon that are in the whole molecule, irrespective of where they sit in the molecule. And those you see also, okay, I injected this person with an exotic compound that differed in the carbon content from their body, just like occurred for Floyd Landis. And I see the signal rise, and then I see it decay, and it's gone in something like half a day to a day. And then we looked at the time series of our measurements, where we're observing the isotopic structure and the multiply substituted forms. And we also looked at deuterium, a rare isotope of hydrogen, and where it was in the molecule. So many different properties. And we observed something that was both comforting, that we're not crazy, we're seeing something that's physically possible. We looked at the isotopes of the full molecule and replicated what we knew had to be true. We saw them rise and fall, just like you would imagine. And then we started looking at the isotopic structural characteristics, and we realized we're seeing another version of this problem that's much richer. We were seeing this sort of chemical equivalent of the bell ringing that I described to you. The isotopic structures of the metabolites, the hormones, they are first jumping to some new composition because of what you've added to your body. And then they're evolving in crazy ways because the entire metabolic system of the hormones in your body, how one hormone turns into another, all of it was disturbed. All of it was dysregulated, and that gets expressed in the elementary reactions, the addition of one atom, the removal of another. As the molecules turn from one compound to another, their isotopic structures change. And we're watching this occur, whether it's the compound you injected or not. You could inject testosterone and look at a different hormone, and its isotopic structure is changing because the regulation is changing, all the chemistry is changing. And we imagined that over the course of 2 days, these signals would disappear because the concentration signal disappeared, the normal isotope signature disappeared. At the end of our study, they weren't back to normal. So the implication is that when you study in this way, you first of all have a highly specific fingerprint, something that can't be faked. Second, you have a very mechanistic fingerprint. It tells you how the system was being dysregulated. And third, a kind of the simplest and most obvious interesting thing, it hasn't gone away yet. It's a persistent change that may be very easy to detect later than some of the more familiar tests. When they stop working, maybe our test will keep working.
Very interesting. So how far out did you look in terms of the timeline?
Just a couple of days.
Okay.
So there's a whole bunch of open questions, and they're the next thing we need to do. And this is the part where the fun exploration, that continues a little bit, but now you have to do the hard part, the hard elbow grease work to turn it into a tool that would hold up in a court of law, that a critic wouldn't be able to poo-poo. So what does that mean? We need to do things like establishing in a more statistically meaningful way what really is the baseline of the isotopic structures of hormones in humans. How variable are they? How do they change in response to just normal things about your diet and your behavior? Second, we need to understand, when is this bell going to stop ringing? We need to study someone over a longer time period after administration and see when do they return back to the normal baseline that we are imagining they will eventually get back to. So this is the next thing that we really need to do. And we haven't figured out exactly when we'll do it, but Lubna is still with Caltech, so the time is ripe for us to try that next step.
Well, I think this is a very exciting time for this project, and I think the implications are huge potentially for the field of anti-doping. So can you maybe comment on how these findings at this preliminary stage even could maybe be sending ripples in the field or having an impact?
I think the most interesting thing about what we've done is that the idea is pretty unusual. The methodology is pretty unusual, but the instruments on which we did the work are everywhere. So that means that we have a whole community, the anti-doping community. There's tons of labs. They're all filled with experts who know all about this problem and are really great lab scientists. They can absolutely do this. They just have to be taught how to do it. So this is an approach that could spread to the day-to-day working anti-doping labs. They could be doing this measurement in months if they wanted to. The question is, how do we show them what we have in our pilot study? And then what do we do over the next year or two to convince them that this is something that's going to be useful and to help them adopt it? How do we teach them how to do this? That's what's exciting to me right now, is how do we turn this from an interesting proof of concept that shows you all the things that are possible? How do we turn it into a tool where you take a sample from a doper that you couldn't catch otherwise and you prove beyond a shadow of a doubt what they did?
Definitely. I think you mentioned some very important next steps for this particular line of research. And I think this has been a real big success story for the PCC's anti-doping research program. But maybe could you share what your experience has been like as a geoscientist applying your expertise to advance anti-doping research?
I find this all very personally stimulating on several levels. Probably the one that is most powerful is it just brings me in contact with new people who have different perspectives, different interests. And this goes back to my camping trips that made me turn into a geologist. I'm maybe not going to turn out of being a geologist, but I love the experience of being with new people, not just chit-chatting at a cocktail party or something, but working with them, watching how they work. I'm working alongside them. We're figuring out how to interact with each other. I find this very personally stimulating. And so that to me is the most exciting side to all of this. And it's just intrinsically fun. This is a thing you read about in the newspapers and then you gossip about around the water cooler on Monday morning. Lots of people like sports. They like to follow these things. They follow the Olympics. It's an interesting social activity that I get to help be better.
Absolutely. And I think the passion for sport and competition is something that drives a lot of people who contribute to anti-doping science. And I really love that you highlighted the collaborations and the people that you work with. I think this is critical in the field as well as many other scientific fields. So can you maybe give us a little bit of more detail on how you collaborate with other stakeholders, whether it's funding organizations like the PCC or other scientists in the work that you do?
Well, the way that I did this was to reach out to Thomas Piper in person, just person to person, pick up a phone, drop an email and hope that this is a person who answers their email, and just be a person who creates relationships. And I think if that's your mindset, so long as you don't mind taking no for an answer sometimes, you are going to eventually find people who are also excited about novelty. They're also excited about working with weirdos from other disciplines. And in the end, human beings do these things. And you're like the door-to-door salesman. If you're not knocking on doors, you're not making any sales. That's my view about how you really make it work. The funding, I'm incredibly grateful that the Partnership for Clean Competition supported the work. And I'm also very grateful just for their existence to plant that seed. When you create a funding agency and program, you're sort of announcing the existence of a community. And so an individual like me can see that and then have the thought, I could be part of that community. So the fact that they exist, that they're visible, that's a super important thing by itself.
Oh, absolutely. Well, we appreciate you sharing your experiences and we appreciate you being part of the PCC's research story here. And I know there are so many unanswered questions in the world of anti-doping, and oftentimes resources are unfortunately a limiting factor. So if we gave you everything you could dream up and feasibility, resources, funding, staff, time, these normal things that hold you back were not a problem, is there one particular question in anti-doping that you would most want to solve, John?
Let me turn it into two. If somebody asks you, do you want 1 scoop of vanilla or 1 scoop of chocolate? You say yes.
Yeah.
So there's two things that I would love to do. One of them is, I described it in unromantic sounding terms, but I really want to know at a pretty deep level, is this something that would actually work in the real world? Like, is there a day you can imagine where I take a urine sample from an unknown athlete and I prove at every level of confidence you want to stipulate that it's either a part of the natural spectrum of human metabolism or this is something disturbed that is a result of doping. I know that that's possible at the end of our pilot study. I don't know that I can just nail it every time. That can only be done by scaling up. You have to do this at a much bigger scale, something that's approaching the scale of study, for instance, drug studies before the release of a drug or other sorts of biomedical work. Human beings are complicated things. Our bodies are very complicated, very diverse. You can't study one or two or three of something and say you've nailed it. You've got to study dozens at minimum before you're really confident you understand what's going on. So I want that to happen. And that would only happen if the ice cream scoop you offered me landed in my bowl. That's a hard one. The second thing, and this is just a personal curiosity, I have a friend who has worked in anti-doping in England. He's like a former Olympic weightlifter, and he's shown up and helped run programs, especially at the big strongman competitions, which they do testing, even though the use of performance-enhancing drugs is not punished within many of those competitions. They still test and see what's going on. So I know from him, just through sort of gossip, that what your intuition would tell you, these people are massively perturbing their bodies using doping agents. And that's their choice. I don't really judge them based on it, but I'm extremely curious as to what it is doing to them. And they represent an extreme manifestation of this thing that we're studying. I just have a natural curiosity. What is going on under the hood? And what does that look like when we observe it using these tools that my lab has invented? It's another manifestation of this instinct I have. Wherever you are, go somewhere you haven't been and see what's there. So that's the part of this that I have not seen, and I can't quite imagine what it will look like. But I can imagine ways in which these technologies, which are designed for these wider purposes, could be applied for all kinds of compounds. The whole purpose of all of these technologies that came out of my lab was to let you very freely wander through all of the world of chemistry, all the tens of millions of molecules that we know about, and study their isotopic structures with all of these questions in mind of like, how was the molecule made and how was it destroyed? And can I forensically separate different origins of molecules, and to do it for anything? So it seems sort of obvious, all of the chemical compounds that people use in sports doping, you should be able to use this tool. You would just use it a little bit differently than we did in the study that I've been discussing. In particular, all the synthetic agents where if you detect it, of course, you know it was synthetic. Human bodies don't make them. But maybe you then have further questions. Am I looking at a single point source that's delivering this drug to everybody? Is it some little batch of something somebody cooked up? Do I have 5 different suppliers? These kinds of forensic questions, you could very usefully apply these tools to separate different origins. And maybe you could think of other uses, like what if I detected a compound, but I don't know how long it's been in their body or what the initial concentration was when they administered it. And then you could also address that question, looking at the isotopic changes over time.
Well, I think these are both fascinating questions. I think just this idea of being able to catch every single doping athlete who is tested with one particular way of testing or analyzing the samples would be huge in the field. And just having this more in-depth understanding of the impacts of doping, I think is really important just from an athlete health perspective, if nothing else.
Yeah. And I understand how grandiose it sounds. The idea, well, you mean there could be one test and you detect everybody with high confidence? It does sound crazy. I'm sure there have been many people who thought that that's what they had on their plate. And maybe I'm wrong, that this could be it. That's why you do an in-depth study. But the possibility actually is there. And it's because this measurement brings something to this problem that is very difficult to bring otherwise. And that is almost boundless complexity. It's sort of like genomics in this way. If I don't get a good answer measuring 10 things, I can measure another 10. If 20 doesn't do it, I could measure another 20. There's almost boundless depth of complexity in the types of things that we can measure if we need to. So that's why I'm optimistic that we might discover this is very broadly useful in this way.
Well, I am very excited about the work that you're doing, John, and I think it has so much promise in the world of anti-doping that we're excited to see where this work goes. And if our listeners want to learn more about some of the things that we talked about today, John, where can they go?
First of all, I would say write to me and write to Lubna Shawar, who is also at Caltech. She's the person really in the lab doing the work. We talk to people all the time. And so long as it isn't 1,000 people who write, we will respond. Second, Google Scholar is a great place to go to learn about the most recent research happening in my group and Lubna's work. And I'm sure Thomas Piper would say the same. 10 years ago, we would have said, go to my webpage, but honestly, that's so 2015. Go to the active online databases and you will see what we did very recently.
Fantastic. Well, listeners, definitely reach out to John and Lubna if you have specific questions, and check out Google Scholar to get the latest information coming from John's lab. And John, thank you so much for joining us on the show today. It's been such a pleasure to chat with you.
Thank you, and to your audience.
Well, listeners, wonderful to have you here with us as well. It would mean so much if you could take a moment to rate our podcast, subscribe, and leave a review on your favorite podcast platform. And please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.